And welcome everyone. We're so glad you're here. My name is Jenny Dunn. I'm the Director of Marketing here at ANRF, and on behalf of our entire team, we want to welcome you to The Bigger Picture, CAR T Therapy and the Future of Autoimmune Care. This is the third and final webinar in ANRF's Arthritis and Lupus Awareness Month series, and we have saved one of our most exciting conversations for you today. I'm truly thrilled to be here with you. I want to take a moment to thank our sponsor, Bristol Myers Squibb, for their generous support of today's program. It's because of partnerships like this that we're able to bring important and independent education directly to you, our community. Before we get started, I just have a couple little pieces of housekeeping. This webinar is being recorded. We'll have it available on our website and YouTube channel in the coming weeks, so feel free to share it with anyone who wasn't able to join. If you have any questions for our panelists, please do type them into the Q&A box at the bottom of your screen. We'll have a dedicated time for Q&A at the end, and I'll be reading your questions to the panel. So if we don't get to yours today, also please do email us at info@curearthritis.org, and we'll follow up on those. We'll also be running a few short poll questions throughout the session. They're such a great way for us to get to know you and to personalize this conversation, so please do participate when you see those pop up. Now, a little bit about today's program. You may have been hearing a lot about CAR T therapy, perhaps in the context of cancer treatment, but what a lot of people don't know is that researchers are now exploring what this approach could mean for people living with arthritis and autoimmune conditions. It's one of the most exciting frontiers in autoimmune research right now, and today we're going to help you understand what it is, where the science stands, and what it can mean for your care. We have two incredible experts joining us today who will walk us through both the clinical picture and the latest research. So I'd love to get us started. And before we start, I want to get to know you a little bit better. So I'm going to pull up a poll. This one is just a question about what brings you here today. Are you living with arthritis and autoimmune disease? Are you a caregiver, a family member, researcher, healthcare professional? What brought you in the door today? And how can we help you learn more? Okay, I'm going to end our poll and share these results. As I expected, we've got a lot of folks with us today who are living with arthritis and autoimmune disease, also some caregivers, some researchers. We are so excited to dig into this and find out more. And so to get us started, I would love to introduce Dr. Irene Blanco. Dr. Blanco is a professor of medicine in rheumatology at Northwestern University Feinberg School of Medicine, where she serves as director of Lupus Clinical Services. Her research focuses on patient outcomes and health disparities in rheumatology, particularly in systemic lupus erythematosus, and she also co-directs Northwestern's Clinical Research Ethics Consultative Service. She brings both deep clinical expertise and a genuine commitment to the patient experience to everything she does. Dr. Blanco, thank you so much for being here.
Thank you. And so I'm going to just move this way, even though Dr. Richardson is with me. So wonderful. And today I'm hoping to give you an overview on CAR T therapy. It's a little centered on lupus because obviously that's my area of expertise, but hopefully we can answer some of your questions a little bit more broadly as how it relates potentially to other disease states as well. Next slide, please. So what is CAR T therapy? So originally this was a treatment created to treat certain blood cancers like lymphoma. The reason why we started thinking about using this for autoimmunity is because lymphoma cells, and we'll talk about these cells a little bit later, is a derangement predominantly of B cells. And B cells is a type of immune cell that is really implicated in a lot of autoimmune diseases. So we figure if a therapy for lymphoma targeting B cells is targeting the same cell that we want to potentially modify or get rid of in an autoimmune disease, it may actually lend itself to be a great approach for our patients with autoimmune disease. The therapy uses a patient's own or donated T cells. So we're going to talk a lot about different immune cells. And so T cells are cells that occur naturally in the body. They are supposed to kill off cells that are being attacked by certain viruses and bacteria. But what if we reprogram those cells for our own benefit? And so that's in essence what we're doing. We reprogram them to attack other targets to help treat patients. And if you look at this schematic here to the right, and we'll get into a little bit more later, But first step is collecting a patient's blood, because for certain trials. For other trials, we use donated T cells. So what happens is that blood is then separated into all the different parts of blood. We take the T cells and we send them off to whichever company we're participating with. There are many, many companies. BMS is one of them, that is looking at CAR T therapy and various other cell therapies for autoimmune disease. They then work to, one of my colleagues talks about how we send the T cells to university to learn a new skill. So the T cells get a certain molecule put on them called a CAR. And what that does is that the T cell now is reprogrammed to go hunt and look for those B cells, particularly deep into the tissues that might be affected by the disease, in order to get rid of those cells that may be causing a lot of inflammation and damage. So the next step, those cells get the CAR put into them, they get sent back. We, of course, make sure that all of the blood products, not only when they leave the site, are tested to make sure that they're very safe and it's actually the T cells that were donated by the patient, etc. The patient typically, even though there are different protocols now, looking at whether or not we even need to use preconditioning with chemotherapies, and we'll talk about that in a little bit. But we typically use, and for the lupus patients on this call, most of them may recognize cyclophosphamide. Heard the name cyclophosphamide. It is a therapy that we use a lot in lupus patients still. We try to avoid it because it does have its side effects. But in the setting of CAR T, we want to make a little room for the T cells. We want to kind of calm down the immune system a little bit so that those T cells have a little room to work and do their benefit. So we give patients cyclophosphamide and fludarabine, and then we give them back their cells. Next slide, please. So why CAR T may help, and I alluded to this a little bit. So really, these T cells are, I like to call them they have VIP access, right? Because presumably they're part of the immune system. When they're autologous or they're from the same patient, right? They have all the markers that show that they belong to the patient. So they get a little extra access to the deep tissues where there may be inflammation. So they are very adept at finding and removing those harmful B cells that are causing autoimmunity and causing inflammation. As they clear out a lot of those B cells, you can think of it as hitting a hard reset on the B cells. So these B cells that, again, occur naturally in the body and are doing what they're supposed to be doing, just doing it wrong. So they're supposed to be activating other cells and also making antibodies. Antibodies help protect us, again, from different infections and help to clear out dead, unhealthy tissue, etc. But in autoimmunity, those B cells are now attacking one's own organs and other vital systems. So by the T cells clearing out those B cells, it clears out all these cells that may be causing inflammation. And then as your natural B cells just start to come back, hopefully they are naive, they don't recognize your own tissues, and they don't cause inflammation. And again, because these T cells are reprogrammed, they can get really deep into the tissues. So heart, kidney, arthritis in the joints, lungs, etc. And hopefully, based on what we're seeing right now, this might lead to a long-lasting remission. Next slide. And so how did we go from cancer to autoimmune disease? So again, this is FDA approved for multiple blood cancers. And actually, there are now a couple of products that are approved for solid tumor targets. And in certain populations, it can really lead to about an 80% remission for pretty refractory cancers. And because there's such a positive signal in these malignancies, in these cancers, we really wanted to see if this could actually work in autoimmune diseases. And again, the goal is typically B cells, but that may not necessarily be the goal for much longer. We may think a lot more broadly in terms of the CARs that we target. We want to reset the immune system to hopefully lead to a drug-free remission. The status of this, though, is investigational. So as opposed to in cancer, where it's FDA approved, so it's licensed, it's ready to go, a physician can just prescribe it and actually have the patient undergo the therapy. You can only really participate in CAR T or receive CAR T as an autoimmune disease, especially if you don't have a cancer that's being targeted by the CAR, through a clinical trial. And there's hope that this will lead to a sustained remission. Next slide. So to dig a little deeper and to really understand the CARs that are available and what they do for both lupus and scleroderma that you'll hear from by Dr. Richardson in a few minutes, we really need to understand what the B cells do. So B cells, just like T cells, are integral parts of your immune system. They're supposed to be there, and they're supposed to, number one, recognize invaders, right? Recognize all of the viruses, bacteria, all the germs really that want to do us harm. And the way they do it is by producing what we call antibodies, right? These antibodies, particularly in lupus, but also in scleroderma, can bind not only to, ideally it would bind to any infection, and clear that out. You could think of it as sort of like the way you would tag things in like a yard sale, right? Like, things to save, things to get rid of, things to donate. Antibodies do that. They tag things, and it tells the body, hey, cause inflammation here. Hey, get rid of this. Hey, clear this out. We want that for germs. We don't want that on our kidney tissue, on our skin tissue, on our brain tissue, on our lung tissue. But aside from just making antibodies and sort of recognizing germs as they come in, they also activate other cells. So B cells in particular act as these antigen presenting cells. So what does that mean? It means that the B cell sees a germ, eats it up, spits a little piece out, and presents it to the T cell and says, hey, I ate this. Is this bad? Is this unhealthy? And if the T cell recognizes it, it says, you know what? Yes, it's unhealthy. I'm going to go and destroy the cells that are infected with that. And this sort of interaction of a T cell and a B cell is a little bit of what we're kind of taking advantage of when we think about CAR T. And also, not only does the B cell present things to the T cell and also other immune cells, but it also secretes something called chemokines, which are inflammatory hormones, which act as a signal to other cells to say, come here. There's something bad here. There's an infection here. There's something wrong. Please come make inflammation, right? So if we think about a CAR T and these T cells getting rid of these B cells, in essence, we're getting rid of the antibodies that cause inflammation in the deep tissue. We're calming down the T cells, so we're not activating them, and we're not really secreting all those inflammatory hormones or the chemokines anymore, recruiting new cells to make inflammation. Eventually, T cells normally, right, because that's actually how we respond to vaccines. So T cells over time recognize the vaccines, make antibodies to protect us in the future from measles, mumps, rubella, chickenpox, you name it, and then become these long-lasting memory cells and plasmablasts. That way, the next time an infection comes back, they can quickly start churning out those antibodies. The problem in autoimmunity is that those B cells, instead of churning out antibodies again against infection, start to churn out antibodies against one's organs. So potentially T cells can even hunt for these more specialized B cells to get rid of them. Next slide. Right, so I kind of alluded to this a little bit. So at least in lupus, how B cells act abnormally, they act abnormally in many, many different ways. Number one, a lot of the antibodies that they cause are harmful, and they don't necessarily serve us a good purpose. They're not attacking the things they're supposed to be attacking. They're attacking things that we don't want them to attack. And for a lupus patient, that's, for example, we see a lot of autoantibodies in the skin. We see a lot of autoantibodies in the kidney, etc. the B cells start to activate a whole bunch of downstream inflammatory cells that really start to then come into sites where there's a lot of antibodies and start making a lot of inflammation. They start churning out these inflammatory hormones, these chemokines, recruiting even more cells. That just makes the whole immune system run amok. And then little by little, as these B cells mature and these B cells are more and more stimulated, you eventually get these memory B cells that keep this autoimmunity and keep this disease and inflammation going. And that's why I want to clear all of these out and hit a hard reset. Next slide. So many of you may recognize some of these antibodies. Some of them are very clearly tied to causing autoimmunity in lupus. Some of them are associated, so we don't really know if this antibody really sticking to the certain tissue may be associated with it. So for example, not every antibody directly causes inflammation in lupus. Sometimes it's indirectly, but we know a lot of them are implicated. So for example, particularly in kidneys, right? So right here, we know that anti-double-stranded antibodies are very implicated in causing lupus kidney disease. We know that C1q antibodies, and there's certain new antibodies that are coming up, but we know that other antibodies, for example, Rho and LA antibodies, are associated with skin manifestations, in particular in lupus patients. And so for us, these are some of the markers that we follow in CAR T therapy because we want to see those levels of autoantibodies ideally go away, but at least go down. Next slide. So to bring it all together, and this is from a great article looking at CAR T, particularly in a lupus patient. So you can see this lupus patient has a malar rash, has muscle and joint involvement, has potentially pericarditis, kidney disease, etc. They underwent leukapheresis, so we took out her T cells, we educated these T cells, we put the CAR in, and we gave it back to her. And what we see in a lupus patient is as the T cells go up here in this green line, the B cells radically go down. And what you typically see and what's been reported is that all of these antibody levels tend to go down, complement levels, which when there's a lot of inflammation, the complement deposits across the body. So we don't want that. We want it to stay in the blood. So the complement levels go up. We see a lot less protein in the urine, so the kidneys are healing. And the SLEDAI is the lupus disease activity index. And we see that lupus disease activity goes down as the T cells go up. So patients are doing a lot better. They have much less autoantibody burden. Their complements go up. So there's a lot less inflammation, and particularly inflammation in the deep tissue, because we have tons of treatments that target B cells in lupus, right? For the lupus patients on the call, I'm sure they've all heard of Benlysta that targets B cells. It doesn't really kill them off, though. It just kind of calms them down. We have stuff like rituximab, and now the new B cell targeted therapy, obinutuzumab, where obinutuzumab, as opposed to rituximab, not only targets B cells in the blood, but also in the deep tissue. And actually, obinutuzumab can get deeper into the tissue much better than rituximab. But nothing can really get as deep into the tissue as the T cells, because again, they have a little VIP access because they're immune cells themselves. Next slide. So which conditions are being studied? Lupus, rheumatoid arthritis, scleroderma, multiple sclerosis, myositis, and many more. Here we're also looking at vasculitis. There are different CARs that are looking at, for example, Crohn's and ulcerative colitis that do the same thing, but for example, the CAR doesn't target B cells. It actually targets a different aspect of the immune system. And so, this therapy is very multimodal. You can switch up the CAR. You can make the CAR a little smarter. You can make the CAR a little better. You can make the CAR attack various different parts of the immune system. And that might be able to help us fine tune exactly how we're modifying the immune system in these diseases a little bit more. Next slide. So what are the risks and benefits at a very, very superficial glance? So potential benefits, let's start with the pros. Number one, we're targeting autoimmune cells deep in affected tissues. Number two, this could be a potential treatment for refractory patients. Again, we have many, many medicines in lupus. Many more are actually on the horizon. Many more than when I started my career as a rheumatologist, where we didn't have Benlysta, we didn't have Safnella, we didn't have any of the meds that we currently have now. It's really to see how far the landscape in terms of medications for our lupus patients, it's been tremendous. But we all know that there are still patients that are pretty refractory that have failed all of these treatments. Because of the deep tissue hard B cell reset that we might get in lupus patients, maybe we could finally have something for our very refractory patients. And drug-free remission might be in sight, right? The idea that a lupus patient may not even need hydroxychloroquine is mind-boggling. And, there are patients now, especially in the early CAR T trials, that have been in remission now for a couple of years, and this is a drug-free remission. Now, we'll talk about some of the risks in CAR T, but there's potentially milder cytokine release syndrome. Again, autoimmune patients maybe don't have as many B cells as, for example, a lymphoma patient, because that's a cancer. So they're churning out these B cells. So maybe because we're not killing as many B cells, there aren't as many risks, but that still needs to be worked out. And there is hopefully immune recovery over time. Now, there is important risks with CAR T, and I'd like to highlight these top three that we really think about. So number one is cytokine release syndrome, but it goes hand in hand with something called ICANS, which stands for immune effector cell-associated neurotoxicity syndrome. So CRS and ICANS. So T cells are killing machines. They are good at what they do. They are programmed to kill off particularly cells that are infected by viruses and especially bacteria that can get into cells. And we're using that mechanism to have them kill off the B cells. And sometimes when T cells rev up, they themselves can cause a lot of inflammation. They themselves are these killing machines that are hunting all throughout the body of an autoimmune patient, of a lymphoma patient as well, because these are side effects that we know of because we've seen them in the cancer spectrum. And so we know that this can happen. So what happens is when the T cells get a little too revved up and they release those cytokines, or so those inflammatory hormones, patients can have fever. They can have low blood pressure. And if we catch it very, very early, we have medicines that help to treat that. And when the CRS, when there's some inflammation in the brain, that could lead to some confusion in patients. That could even lead to seizures. So something that we do is in CAR T patients, we monitor them very, very closely. If there's any signs of fever, if it's a very mild, low fever, we just watch, maybe some Tylenol. But as soon as the fever hits a certain threshold, especially here at Northwestern, we give anti-inflammatory medicines to decrease CRS and to make sure that it doesn't progress, because we know that the sooner we treat it, the better the patient does. Many protocols now require patients to be on seizure medicine prophylaxis to make sure that you do not get confused or have a seizure. And we monitor you while you're in the hospital or while you're with us, asking you these questions over and over and over again. Do you know where you are? Do you know what date it is? Can you write this sentence for me? That way we find any sort of confusion or memory impairment very, very early. And so we can actually really mitigate CRS and ICANS in our patients. Can it progress and be severe? It can, but again, early treatment really helps to prevent bad outcomes. B cell depletion really does lead to infection risks, and B cells really talk to other immune cells. So when B cells aren't around, other immune cells can go down as well. So while the B cells are recovering, we need to monitor patients very, very closely for infection, but we also give them things to protect them from infection. So patients early on in their CAR T therapy will be on antibacterial antibiotics, will be on antifungals and antiviral medicine to make sure that you don't get an infection. Again, many protocols now require conditioning chemo before the infusions. We'll see whether or not we need it, because again, these aren't lymphoma patients. And again, there's no long-term safety data, and this is only available through clinical trials. Next slide. So what does participation look like? If you're interested, please find a site, talk to your doctor, you'll reach out. You go through a screening process, which can be anywhere from a few weeks to actually up to a month, where potentially you may get some studies done, blood tests, et cetera, to make sure that it's safe and you're eligible for the trial. Then you undergo apheresis, your T cells come out, we send them to the company where they're manufactured, the CAR gets put in. You get the short chemotherapy, you get the infusion, and then we monitor you. The most critical is in the first 30 days, but really here at Northwestern, we monitor the patients very closely after CAR T for pretty much the first year. Next slide. Questions to ask your care team and what to think about. So number one, is CAR T being studied for one of your conditions? Number two, what would you need to do to change your current treatment? So for example, in the lupus trials, we need to bring down some of the immune system because we need to have T cells to modify, right? If you're on too much mycophenolate, we may not be able to collect T cells, and that could be an issue. Number three, what are the most important risks for your situation? For example, if you have a history of seizures, how would the care team mitigate that? How would they ensure that they monitor your safety? Are you a candidate, right? Like there may be trials available for your disease, but you may not necessarily be a candidate. Is travel required? So, we do such close follow-up that we really need the patients to be somewhat local, not 100% local. Some people could fly in and out if that's something that they can actually do. But ideally, patients are within the general vicinity so we can stay on top of them and make sure that they don't have any critically poor outcomes. And if eligible now, what might change? If you're not eligible, like how potentially could you be eligible in the future if you're interested? Next slide. And so what's the evidence showing so far? We have actually now there's many patients. And so there's a new meta-analysis that shows that there's well over like 250 patients that have been treated for autoimmune indications with CAR T. There's many trials that are active worldwide. And again, the longest reported remission is about 4 years of drug-free remission in a lupus patient. Next slide. So what does this mean for you right now? CAR T is investigational, i.e. only available through clinical trials. You don't necessarily need to wait for your current treatments to fail. So even if a CAR T trial is not right for you now, maybe there's a different clinical trial that might be right for you. Not everyone is eligible, but definitely speaking with your providers can give you a sense of what might be next steps. And you can always look at clinicaltrials.gov to see if there are any trials close to you. Next slide. And with that, I will leave you with Dr. Richardson.
That was awesome. So Dr. Blanco is one of the leading experts really in the country. So that's really quite a pleasure. And it's lovely for me to hear that too. So I'm Dr. Carrie Richardson. So I'm a rheumatologist. I specialize, like Dr. Blanco, I specialize in the Scleroderma, also called systemic sclerosis, but I see a variety of autoimmune conditions, including myositis. I'm a co-investigator on 3 of our Scleroderma clinical trials right now that are actively recruiting. Just disclosures, I did help design one of the trials for Cabaletta Bio. So that is part of my disclosures. That was a few years ago. And I'm also a consultant for Priovant Therapeutics. Next slide, please. So Dr. Blanco gave an amazing overview of what CAR T cells are. To me, immunology, especially thinking about it, it is, I like to make it even more simple, right? So, all of our immune cells, they're like ships at sea, and they carry flags. The way that ships at sea might tell you what country a ship is from, our cells have flags on the surface, and those are called cell surface markers. The B cells in our body have these flags called CD19, and some of them, the old B cells, have flags called BCMA. And that just tells your immune system, hey, I'm a B cell. That's who I am. It doesn't mean bad or good, but we have drugs that specifically identify the flags on these B cells. And we have drugs, Dr. Blanco mentioned rituximab, obinutuzumab. There's other what's called monoclonal antibodies that we know we've been using that target these B cells, and they destroy them. But we know that the drugs that we know that they are effective for certain conditions, but they're not cures. So the drugs that we already have that target these cells, they can't get into the tissue. They're too big. They can't get where they need to be. These B cells hide in the body in places that only another cell can get. So the point of the CAR T cell is actually to create these T cells that can find these flags on these cells and say, hey, this is a B cell. I'm going to get rid of it. So there is a hope, and I have that hope too. I have friends and family who've been affected by autoimmune disease. My hope is that the next generation will have no autoimmune disease. We will be able to cure people and say, come back in 5, you're in remission, right? A real remission. Because right now, we're not really able to say that with the diseases that we essentially say you're stable, you have a chronic disease, you might meet quote unquote remission criteria, but it's not the type of remission that we want. We want really to say, hey, you're good. So the CAR T cells, the hope in them is that by going after these B cells, you can start over. So you wipe out the B cells and the new ones come back fresh as if they're like baby B cells, like they're like when you were born, because most people are not born with autoimmune disease. And so far, there's some data that supports there's still reason to hope for that. Now, how long will it last? We don't quite know. There is a risk too that, there may be some people that even the B cells that they're born with might be prone to develop that autoimmune reaction again, even if you start over. So, my hope is that ultimately over time, as the science develops, we will see a true lasting remission, a true reset. But I'm cautiously optimistic. Next slide. CAR T cell goes back even longer than this. This is a snapshot of CAR T. So as Dr. Blanco mentioned, these therapies were actually developed for blood cancers. And so they have been total game changers for lymphoma, and they can do things that other drugs cannot. So really the science goes back to about the 1980s. Carl June, who's here on the right, who's at UPenn, was one of the pioneers in CAR T cell therapy. Georg Schett is one of the pioneers in CAR T cell therapy. So he's Austrian, one of the pioneers on CAR T cell therapy for autoimmune disease specifically. So he's been really instrumental in helping to move that field forward. But the two scientists have been critical to development of these therapies. And so the point of this timeline is to say that when I was, doing, consulting on this trial for Cabaletta a few years ago, most rheumatologists didn't even know about this. They were skeptical of the concept of CAR T, right? So CAR T did not exist, really in any functional form in 2022. The studies were just coming out, and now you see this explosion of CAR T cell trials, which is exciting. It does make me a little nervous as well, because here at Northwestern, we have amazing expertise with one of our autoimmune immunotherapy experts, Dr. Georges, but there are a lot of risks involved with these trials. So you really need an expert center that knows what they're doing, because there's a lot of, there's a lot of wonderful companies that are working in the space, but it's a little bit like the Wild West right now, where there's a lot of different products, different companies, different protocols. And so you have to be very careful about making sure you choose a place with the right expertise to know that you're a good candidate for this and that the product is something that is going to be helpful for you. Next slide. So as Dr. Blanco mentioned already too, there's lots of data that's accumulating. So there was a good meta-analysis in Lupa, which is just a collection of different studies all put together, which is considered high quality evidence to put a number of studies together that showed that, I'm going to say improvement in lupus. So they met about 70% met remission by what's called it's a measure of remission in lupus that still allows you to be on prednisone, which many of us might say that's not a real remission, right? If you're still on medication, is that a real remission? But about 70% did meet remission by that criteria where they were still permitted to be on some prednisone. And about half did have an inflammatory reaction to the CAR T. That's called that cytokine release syndrome, where people can get fevers and need oxygen. And most of those cases were fairly mild. Occasionally, that can be more severe. Lupus is where we have the most data. Scleroderma, systemic sclerosis, which is the disease that I treat, is probably a far second in terms of data. So there is good data actually supporting that in systemic sclerosis, where people get hard skin. I don't know if anybody on the webinar has systemic sclerosis, but systemic sclerosis where people get hard skin and they can get hardening of the internal organs. So what they had seen is that the hard skin, we measure something called the modified Rodnan skin score, that dropped by about a third in the patients. And that was often within the first 3 months that you saw that drop. Now, these are open label studies in scleroderma. So when you give a therapy, you ideally want to look against something, where you don't know that where the person who's measuring doesn't know what the person who received the therapy got. So there's always some bias there because, the person doing the scoring, everybody knows that this person got a CAR T. However, in these studies, they did also show that it probably was not just observer bias where somebody's actually biased towards wanting to believe that it's a good outcome. They actually showed that there was a drop in some of the blood markers of scleroderma and some of those blood markers of autoimmunity as well, which is really exciting. And that has been seen in lupus too. So there is, so these are really, there is a biological effect to these drugs that can be measured. Myositis has even less data. That's emerging evidence. There are some case reports showing that the patients with myositis, which is muscle inflammation, and people with this can get skin inflammation or lung inflammation. There's some data supporting that they can get better with this type of therapy. Rheumatoid arthritis, this type of therapy is being explored, and there's different ways that the CARs are actually being used. There's ways that there's companies using the CARs to try to enhance other therapies. So this is rheumatoid arthritis is still emerging data. All this is emerging data, but some is more emerging than others. Next slide. So this is only a fraction of what's next. I didn't even want to go. There's this whole category of products called BiTEs that I'm not even putting on the slide. These are actually basically like linker autoantibodies, which are proteins that are shaped like a Y that will connect two cells, a T cell and a B cell, to actually destroy that B cell. So that may be something that people are going to see. So that's just, we don't need to go into that right now. I didn't want to put it on the slide because it's a whole other topic. But at some point you might see people looking at things called BiTEs, or you might hear about BiTEs, which are bispecific T cell engagers to try to do the work of a CAR T without actually creating a CAR T. But in terms of CAR T, what's coming next? So when we think about CAR T, we think about allo and we think about auto. Auto means self, allo means other. So the types of CAR Ts, many of the trials are auto CAR Ts. So autologous CAR T, so they actually take the person's own cells, harvest them, and make them into a CAR T, which actually can take time because you have to harvest the cells, and then you have to create them, and then you have to ship them back, and then infuse them. So if somebody's really, really sick, it can take sometimes too much time to deliver that therapy. Allo CAR Ts, allogeneic, so the already pre-made CAR Ts, you may be seeing some of those products become more in use because theoretically you can make them, and then when someone needs them, just ship them out. So they may reduce production time. And then there's also different CAR T targets. So we've talked about something called CD19 and BCMA that are present on B cells. Those are the primary targets of a lot of the clinical trials we see. But over time, there are many, many flags on many immune cells in the body. And we may be seeing that different CAR Ts may be starting to target different flags on those immune cells. There's CAR T regs, which the goal of those is to just try to go into the area of inflamed tissue. And you may see this in the neurologic space and things like MS, where the CAR T regs are really trying to be targeted towards cells like nerve tissue to go in there and calm down the immune reaction. There's in vivo CAR T that is very early. So these are actually, it's called lentiviral vectors, slow virus that actually goes in and creates the CAR T cells in the body. These are preclinical studies, meaning animal studies. So that data was presented a few years ago at our abstract session, is very, very early. But that is something that could be something happening in the future. One of the things that's being explored right now is chemo-free regimens. So right now, we all love CAR T. We love the idea of harnessing the power of the immune system to change one's own autoimmune reaction. However, it still does require some traditional chemotherapy so that your body doesn't reject the CAR Ts. And so for some people, that may not be ideal. So someone who is planning to have children in the future, some of the regimens can potentially impact fertility. So ideally, we would love to have a regimen that doesn't involve that chemo. And so that is something that's being explored in different trials. Are there ways to tweak the CAR T so that they can survive the patient's own immune system in order to expand and do their job without the chemo? And then there's alternative CARs, and Dr. Blanco mentioned that as well, but there's CAR NKs, people are developing CAR macrophages. There's many, many immune cells that you can make into CARs, and they have different properties. So, CAR NKs don't last as long in the immune system, but some people hypothesize that maybe they're a little bit more gentle. So I think, go ahead, next slide. Yeah, so in terms of, there's many, many things that we don't know. So, right, so how, what does remission really mean after CAR T? Are we going to have to change what we think? In scleroderma, I don't typically tell someone you're in remission, right? Because it's just not possible right now. We say stable, but in the future, will there be a different definition of remission for these conditions because we have better therapies? How long can we remain off of other medications? And then who is the right person? A lot of the clinical trials that we do in scleroderma are really targeted towards early disease. So people who've had this for 20, 30 years, for many of the trials, not all, for many of the trials, they're not eligible. There will be trials where with expanded eligibility criteria where you might be able to have had it for longer and have had more damage accumulate and still be eligible for the trial. Right now, you have to be sick, but not too sick. So you have to be in that sweet spot where the risks of CAR T are worth it, but it's not so risky to you that it would hurt you. And so finding that, I think the question is, where is that sweet spot, right? Where is there an optimal time to do this? Should we be doing it earlier? Should we be doing it later? There's questions about if someone's received other treatments like rituximab, how do those treatments impact the effectiveness or the safety of the CAR T in the future? So these are questions that need to be answered. And then how can we speed up the process? So some things like the in vivo CAR T, the allogeneic, those are ways that are kind of being explored to see if we can speed it up, because it takes about 2 weeks, from the time someone's blood is harvested to the time the cells get back. For some people, that's too long, right? And so we need to have options to potentially speed up that process. So who's most likely to benefit? Are there people who are going to do better with this therapy than others? Are there people who are more likely to be harmed, who shouldn't be doing this therapy? And then what if you relapse, right? Or what if you just don't get enough benefit from it? What if, let's say, you get halfway better with the CAR T, but then you plateau, you don't get any better? Could you get another treatment? What would be the side effects of that? So I think those are questions. What other medications would be safe after you get CAR T? And then look at the long-term, the longest lupus patient, 4 years, right? And so that's great to have that data. But what about 10 years? What about 15 years? And that will come with time. Okay, go ahead, next slide. So there are larger trials underway. There's trials that are getting close to getting to what we call to market, where the FDA approves the therapy and says, yes, this is good to go, and someone can get it outside of a clinical trial. I think in the even longer term, you might see these products of like off-the-shelf products where, you know, there's rapid access right now. Doing these CAR T-cell trials, one of the limitations is there is a list of people who need CAR T. So when people get enrolled in a trial, they actually have to wait for the other person to finish their CAR T in order to get into the trial, and then it has to be manufactured. So there is, it's a safety reason that we do this in this way, but are there ways to accelerate this while still remaining safe? Personalized treatment with the trials that we're doing now, we're going to learn about who's most likely to benefit, what are the risks. And then I think that's the question is, in the long term, is this going to be an option for more people? I know a lot of people right now have a lot of, you know, they go and ask about the CAR Ts, and it's very disappointing because they want to do the CAR T, they're sick of having autoimmune disease, they're sick of being on these medications, but then they're not eligible for the CAR Ts. When we look at these trials, the CAR T trials will have about 50 inclusion and exclusion criteria, meaning you've got to meet 50 check boxes and say, yes, you qualify for all these. That's very typical. So it's very common for people not to check all the boxes in a clinical trial. A clinical trial, they're looking to make sure it's safe, make sure the trial is successful, make sure they have the best chance of getting the result most accurately. And so they want to make sure everything goes well. Over time, though, as we learn more about these, there are people who may not have been eligible for the trials, but they might be eligible in the future. Is this over time? So we'll often see that access to an effective therapy will expand over time as we learn more about it. So that's the question, is this going to be something that we'll see in greater use compared to our traditional therapies? Next slide. And then with that, it looks like we have time for Q&A.
Thank you both so much. That was just a wonderful way to connect the science for us. I'm sure you've seen we have a ton of questions coming in the Q&A, so I think we'll head right in there. Please do feel free to add more of your questions. We will get to these as many as we can today, and we'll follow up with them after the webinar too. So the first question we had come in is, how often would a patient need to do CAR T therapy? I think this is really a question about what this treatment looks like in practice. Are there any suggestions for working with your doctors or insurance companies? So maybe, Carrie, you could take that one, Dr. Richardson.
Yeah. So CAR T right now is once. So we don't know if someone can be retreated. That is still an open possibility in the future, but I'm not aware of any protocol for repeat CAR T that's come across my desk. So right now it's a one-time therapy. And with the clinical trial typically covers the expenses related to the actual treatment. So there are certain expenses that they may not cover, like if there's some adverse event, you know, and you're hospitalized for some other reason during the clinical trial, that it would typically be where insurance kicks in. But right now, under a clinical trial, if you are eligible and appropriate and qualify for a clinical trial, the expenses of the actual treatment are covered by the company that's sponsoring it.
Right. And what does it look like? I mean, if you can just give folks a sense of what does the actual treatment look like? How often do you have to come into the office? Things like that.
It really depends on the protocol. So, you know, depending on the company, so at Northwestern, we tend to be generally very conservative because we want to just sort of stay on top of these patients, right? So, for example, a patient will be hospitalized for 2 weeks. So to get the pre-treatment chemotherapy, that's actually outpatient chemotherapy. But for patients that live far away, they may want to stay close, right? So, for example, we've worked with different sponsors to have hotel stays be paid for patients, so that way they can get their pre-infusion chemo very close and not have to like travel 2-3 hours back. So they get 3 days of the chemotherapy with fludarabine and cyclophosphamide, and then they get admitted to the hospital for the infusion. The same day that you get admitted, you get the line placed and you get the infusion of the cells. And then we pretty much stare at you for 2 weeks and make sure nothing goes wrong in terms of CRS or ICANS. And it's literally staring at you. So like patients by like day 10, they are, just wandering the halls going, yep, I'm still okay. And we're like, knock on wood, your mouth to God's ears. But then we have you again, you can go home if you live very close to the hospital. We say that's usually within an hour range of the hospital, including traffic, because we have Chicago traffic, right? So an hour by distance is not an hour by time, right? In all reality. And so then we, again, work with sponsors that if the patient lives outside of that catchment area, they actually stay at a close by hotel for them the next 2 weeks. So we can monitor them, and then we have them come back every month for the first year, and more if something happens, if they have an adverse event, if they have an infection, etc. But then after that first year, it's every 3 months. And then depending on the protocol, maybe every 3 months for the next 2 to 3 years. But all the CAR-Ts want patients to have some sort of touch point with either the company or the trial location for upwards of 15 years. Because, again, we don't necessarily really know long-term what this looks like in patients.
Yeah. Thank you. That's so helpful. Hopefully that gives folks a sense of what it would actually be like. For you, Dr. Blanco, we had a question come in. For lupus patients experiencing drug-free remission, do B and T cell numbers stay within normal ranges over time, or do we see a return of autoreactive B cells?
That's a great question. I don't think we really know. So, we're killing off all your B cells. So in the immediate time of CAR-T, your B cells are going to plummet and they're going to be very low, which is why we give you those antibiotics, antifungals, antivirals to make sure you don't get an infection, knock on wood, right? And we tell you, wear a mask, wash your hands, stay away from anybody, don't buy anybody that tells you, oh, it's just allergies, get away from me, right? So on top of it, because B cells stimulate T cells, those numbers tend to go down as well. And we monitor them. That's part of the thing that we look at every single month, looking for those cells. Then as those B cells and T cells start to come up, then we peel off all the antibiotics. And ideally, we get back to completely normal numbers. In the lymphoma patients, and so far in a small number of the autoimmune patients, we have seen the B cells take a little time to come back. And for example, some B cells, in some patients, we may need to revaccinate them. So we look at vaccine titers, like we sometimes have to revaccinate patients to their childhood vaccines because their vaccine titers have gone down because the B cells are just taking a little longer to come back. And it's really going to depend on the patient and potentially some of the medicines that they may have gotten prior to the CAR-T that have maybe made changes to the bone marrow, things like cyclophosphamide.
Yeah. We're seeing a lot of folks ask if they want to get involved with something like this, where should they go? Dr. Richardson, is the first stop your rheumatologist?
Oh, absolutely. And
Right.
I would say, and usually too, I would say first stop is rheumatologist is a great stop. And then often, for me, I have a lot of the local rheumatologists on speed dial. I know a lot of people do not see a rheumatologist in Chicago or in Philadelphia or one of these major CAR-T centers, and that's fine. But a lot of times I will have a doc-to-doc conversation with the rheumatologist. We will review the records. So sometimes that's what we do too. If somebody we know it might be a candidate, we'll typically bring them in for a visit and we will review all of the data and make sure that they're appropriate. I would definitely recommend, if you're going to do something like these are risky therapies, go to a center where they have expertise in this type of therapy, not only just in giving it, because it's not about just giving it. Anybody can give it. It's about managing the side effects. It's about making sure the safety is there and also making sure that the person who's going in is appropriately, is the right person for this trial, that their disease activity is appropriately measured. So some of these things, that's where I think the expertise really comes in, is matching the right person to the right trial. And we do have other trials, right, that are not CAR T.
Right.
There may still be a role for those therapies that are not quite this extreme. We have stem cell therapy, we have CAR T cell therapy, we have other
Mm-hmm.
clinical trials, we have standard of care therapies. So we kind of have these levels of like who is appropriate for what level. So you have to make sure that you are at the right level of, having disease activity and also being, safe for it, that you're being matched
Right.
with the right kind of therapy.
Right. So I would say that's a great point. You can go to your rheumatologist, other places to look. You can always check on websites like ours at curearthritis.org, and of course clinicaltrials.gov, I think is what that one is, to look around for what trials might be available. And then like Dr. Richardson said, you've got to work with folks to see what might work best for you. I think we're gonna have to close the Q&A here for today. Please keep a lookout though, folks. We're gonna do some follow-up pieces on this just to make sure we get all your questions answered. We know this is such a hot topic, and we cannot be more grateful to you, Dr. Richardson, Dr. Blanco, for your time today, for your insight, your expertise. This was such a wonderful primer for us on what CAR T therapy is and how it might work for people in our community. So we thank you. Before we go, I want to have folks answer one last poll. You'll see it's our knowledge check here. How confident do you feel talking to your doctor about CAR T therapy or even just emerging treatments after today? Do you feel more confident? We hope that today really kind of prepped you to get into those conversations with your doctor. And I think you'll see, yeah, a lot of us do feel more confident after being here today. And we're really glad to see that. So a couple of things as we close up. I want to say again a huge heartfelt thank you to Dr. Blanco and Dr. Richardson. Your expertise and generosity with your time today just truly means so much to us and to our community. Um, we also just want to give another thanks to our sponsor today, Bristol Myers Squibb, for making this program possible. A couple things before I let you go. This webinar will be available on demand, so we'll send you a link in your follow-up email. You can rewatch it, share it with somebody who might need this information. That would be wonderful. If today's conversation inspired or helped you, I hope you'll consider supporting ANRF. The research we've talked about today is made possible by donors like you who believe in what we do. So please do visit us at curearthritis.org to learn more and make a gift. And then finally, we just want to thank you again, all of you, for being here. Please follow us on social media to stay connected and check out all that we've shared this month. We've had 2 other wonderful webinars, and today was no exception. Thank you all so much. Goodbye for now. Please do take care of yourselves.